Welcome, everybody. Hi, Fabian. We have a lot of big names today here. That's cool. Young is an audience. I think today I'm letting Rainy introduce our speakers. So I'm just going to directly pass the microphone
Thank you very much. So this is spontaneous, so if I get stuff wrong, please correct me. So yeah, it's a great pleasure to welcome Jon Wolpaw to this series. Jon, probably everyone knows him, but he was one of the pioneers. I really had the pleasure to meet him early days together with Teresa and other people when I joined the Graz team. And he was always a strong supporter and a strong pusher into make really BCI technology for the patients and keep us, the engineers, maybe grounded with really practical applications for really the translational aspects of research. So, sorry? Okay, no, I heard something. Yeah, I guess that's my very, very informal introduction. So he's really a big player in the field, one of the pioneers who really drove the whole area of BCI together with a few other people. And it's a really a huge pleasure for us to have you here, Jon, to give up the presentation of your newest ideas, share them, discuss them. So I'm really looking forward to see the recording because I have to run to another other meeting then. But so very welcome. And if you want to add something more detail, I can remember. It's just he's one of the big player in the field who really drove the development and the idea and made BCI basically what BCI is nowadays. So really a big pleasure to have you here and a big honor. Thank you.
Right. Yes, so thank you very much, Rainy. It's really great to see you again. It's really nice to be able to talk in Essex, particularly at this time. It's a really very exciting time, as I'll try to convey for neurorehab and for technology. I've certainly profited a great deal from my interactions, certainly with you and other engineers over the years. I hadn't previously known how valuable engineers are. But I certainly appreciate that now. And it's been great to be able to work with a lot of engineers over the years. It's nice to meet Anna and see Sebastian again. I'm glad to be able to be here. And I'm going to talk about 21st century neurorehabilitation, as it says in the heading, new science, new strategies, new expectations. I hope I can convey the excitement of it. I want to talk about how this has come about through the past 50 years. Really over the past 50 years, when the hardwired CNS of 1970 has become the plastic CNS of today, in which change is the rule, not the exception. For people who weren't around back then, which I realize is most of the people here, it's really hard to comprehend, to understand the hugeness of the change. And it really affects rehab tremendously. This transformation has made rehab research one of the most exciting areas of neuroscience due to the scientific insights it produces and the novel therapies it pursues. It used to be 50 years ago that it was assumed that you had damage to the nervous system, you were stuck with it and you couldn't fix it. And all you could do was work around it. And so rehab research became little more than building better wheelchairs. It was it was a real backwater research-wise, and that's completely transformed now. It really is leading in many ways the advances in neuroscience, and it's extremely exciting both for scientific and clinical reasons. We now know, and that's what the major scientific, the core scientific part of that is, we now know that the central nervous system changes continually throughout life, which we didn't know 50 years ago. There's neurogenesis that occurs, gene activation, neuronal properties change, LTP, long-term potentiation and depression occur in synapses, glia change, dendrites change, hormones change, there's sprouting that occurs, there are even vascular changes. All these different kinds of plasticity are going on in all of us all the time through our lives. And somehow they get combined and maintained to allow us to acquire and keep all the skills we acquire through life. Somehow all this plasticity gets synchronized so that we acquire and maintain all these useful skills. This goes all the way from flexion withdrawal reflexes, which are actually skills that are acquired in utero and early in life, through standard skills like locomotion and speaking to the most sophisticated cognitive and athletic undertakings. So it's a new concept of how this comes about, a new realization of how this comes about. It introduces lots of possibilities, and at the same time, it makes the science of this much more complicated than we thought it was 50 years ago. So the challenge for rehabilitation is to take all these various kinds of plasticity and somehow produce recovered skills. How do we take all this plasticity, which in the healthy nervous system acquires and maintains skills? How do we take that plasticity and use it to produce recovery after a stroke or a spinal cord injury or another trauma has occurred? So there are really two kinds or two classes of therapeutic approaches, which are now both yielding a good deal. First, you fix the disability. And perhaps the best known current example of that is functional electrical stimulation to suppress tremor, for example, in people with essential tremor or Parkinson's disease. This is an ongoing therapy. You keep it in place and it fixes the disability. And it allows you to function much better. The other approach is to try to fix the pathology. And this would be a transient treatment that you just, for example, if we had treatments to repair, as we're perhaps on the verge of producing in humans, that we've produced in animals, to repair pathways damaged by spinal cord injury. That you do that, and the idea is to restore healthy function, to return the nervous system to functioning the way it did before the trauma or the disease. And I'll be talking mainly about that. I mean, clearly both of these are important and will be important for a long time. But this is really the most exciting, kind of approach, and the one that recognizes the new science that we've developed over the past 50 years. So the question is, we've learned a great deal more about how skills are acquired and maintained in the healthy CNS. And that's what I want to talk about for a little while, because this is the background to what I'll be talking about later on in the talk on rehabilitation. And if people have burning questions that they need to ask now, I'll be happy to answer questions in the course of things or at the end, either way. So there are really we've had two crucial insights as to how skills are acquired and maintained. First, we now know that a skill is not exactly the same every time, even when it's performed by an expert. This is Nikolai Bernstein, who many of you may know. He was a Russian physiologist in the early and middle 20th century, made major advances in understanding sensory motor control. He worked in very difficult circumstances. He had two major disadvantages working in the Soviet Union at the time. First, he was a Jew, which made his life more difficult. There were positions that he was not eligible for because of that. And the second was that he didn't always agree with Pavlov, who was viewed then as infallible in the Soviet Union. Nevertheless, he thrived through his life and contributed a great deal. And this is one of his insights, one of his demonstrations. So this is an expert who's using a mallet to hit a chisel. And he's hitting the chisel at two different points. But the import of this figure is that the trajectories for the two different points, for each of the points varies. It's not exactly the same every time. What is exactly the same, so the trajectory varies. What doesn't change is where the hammer hits. For this target, it always hits there, no matter how much the trajectory varied in between, or what the exact course of the trajectory was in between. And here's target 2. It hits exactly there. This is the key feature of the trajectory. That's really all that matters about the trajectory. It defines the trajectory as satisfactory. So skills have key features that define them as satisfactory. They aren't exactly the same every time. The muscle activity, the kinematics can vary. Then the second major insight is that the substrate of a skill is a distributed network. Back in 1970, people could have the notion that the brain was like a computer, that you had the hardware, which was most of the brain, and then you had software, which was squirreled away in special places, the software for locomotion, the software for speaking, and that got downloaded into the hardware whenever it was needed. We didn't really understand how that happened or exactly the details, but it was a very simple concept. It was easy to understand that as the thinking of the brain as a computer. That's not correct. We now know that the substrate of the skill is a distributed network. There really is no hardware. The skill is distributed throughout. And this is a study from Doyen's lab in Montreal, an fMRI study of the development of the skill of a simple finger flexion task, a finger flexion sequence task. And the colored areas show where the plasticity is occurring that underlies this skill. So there's plasticity in cortex, there's plasticity subcortically, there's plasticity in cerebellum, and there's plasticity in spinal cord, part of the spinal cord that controls the fingers. So plasticity in all these areas makes overlapping and independent contributions to the skill development. So this is a distributed network that produces this relatively simple skill. We've been looking now for a number of decades at an even simpler skill, operant conditioning of the H reflex. The H reflex is the electrical analog of the knee jerk. So if you stimulate here, in an animal, we do it with a stimulation cuff. In a human, you do it from the skin. You produce input, which goes in synapses on the motor neuron and mostly monosynaptically, and goes out and produces an H reflex, which was described by Hoffman now something over 100 years ago. Here you see the H reflex here. In a rat, it's occurring at about 6 to 10 milliseconds. So this is the earliest and simplest response of the mammalian central nervous system. It's totally spinal, but it's under the control of descending control pathways from the brain. And because of that, we've shown and others have confirmed that monkeys, rats, mice, and people can gradually change H reflex size when they're rewarded for doing so. And this shows the phenomenon as described in the rat by Shenyan Chen in our lab. These are rats in which the H reflex is trained up over days. This is a 50-day period, or trained down over days. And this is Aiko Thompson, who is a postdoc in the lab, demonstrated the same phenomenon in people, the gradual increase with being rewarded for bigger reflexes and the gradual decrease being rewarded for smaller reflexes. And this change is still present here several months later. So this is the simplest, really the simplest behavior of the mammalian central nervous system. And this is our current understanding, which is obviously very imperfect and partial, of what's happening, what the underlying events are. These are the people in the lab who've done this: Shenyan Chen, Yu Wang, Jonathan Karp, Lu Chen, and Yi Chen, over 30 years. What we believe is that the reward produces input to the inferior olive, which produces plasticity. This is change shown by the pink oval in the cerebellum, which produces plasticity in sensory motor cortex, which produces corticospinal tract activity, which produces a small decrease when it's in place. The corticospinal activity produces, this is decreasing the H reflex. When we're rewarding a smaller H reflex, you get a small decrease here from the CST activity. And this, over days, this gradually produces plasticity at a multitude of places in the spinal cord, which is responsible for much greater decrease in the H reflex. So when we first started to do this, I at least assumed that the change would be just here. It would be very simple, it would be very focal, and it would just affect the H reflex. But that's not what happens. You get a hierarchy of changes throughout the nervous system, the brain and the spinal cord. The plasticity in the brain guides and maintains the plasticity in the spinal cord. So here are two networks of very simple skills. This is a network that underlies the finger flexion sequence learning I talked about from Doyen's lab. This is our simpler skill of a altered H reflex and the distributed network of plasticity of changes in neurons and synapses that underlies that. So we have new knowledge from this. First, we now know that the CNS is plastic through life. The skill details vary. Key features are maintained. The skill can be different from one performance to another, even by an expert, but the key features are maintained, so the skill stays satisfactory. The substrate of a skill is a network of neurons and synapses that goes from the brain down to the spinal cord. Now, these networks inevitably, and this is really important, these networks overlap. They share neurons and synapses. If you take one neuron or one synapse in the cerebellum or in the spinal cord or in the cortex, it isn't devoted to one skill. It's serving all sorts of skills. So that brings up a very important new question. How do they maintain how do all these networks maintain their skills in the changing central nervous system? It's continually changing. You're learning new things. Your nervous system is changing in other ways as you get older. How do these networks maintain their skills? There are changes occurring throughout the network. This requires, I believe, a new paradigm. And the basis of the paradigm is that these networks have two special properties. So they have an individual property. Each of these networks changes continually to maintain the key features of its skill. Many studies over recent decades have shown that skills are continually changing as they're performed. They're continually being optimized, errors are being corrected as they're performed. And this is happening in all of us as we walk. That's continually being adjusted to maintain satisfactory walking. So key features for locomotion, for example, are things like symmetry, posture, balance, acceptable metabolic cost, etc. The EMG and the kinematics may change, but the key features are maintained. This network is really like a person with one purpose in life, maintaining its skill. So there's also a collective property. All the networks are changing concurrently. They are changing the neurons and synapses that they all share. Thus, the aggregate process is essentially a negotiation. The networks are negotiating the properties of the neurons and synapses they all use. They keep the central nervous system in a negotiated equilibrium that maintains all their skills. This is the healthy nervous system. This is similar to a Nash equilibrium as defined in game theory, in which each of the players can't really better his or her position by any change because somebody else will make a change which will cancel it out. So they're all reasonably happy. So these networks overlap. They share neurons and synapses, as I said before. And we've had a very graphic example of this in a study just published last year by Yu Wang in our lab. We're looking here at a motor neuron in the spinal cord. This is a rat, this is a soleus motor neuron, but you have the soma of the motor neuron and you have the axon initial segment. This is where the axon potential is triggered. This is really the final gatekeeper that decides whether or not this motor neuron fires. So this is where everything that's going on in the nervous, the final gatekeeper for everything we do, the axon initial segment of the motor neuron. This determines whether anything happens out there in the world because of what's going on in our brains. So this is the normal situation, and here we have it producing the H reflex. we've looked at what happens to the axon initial segment when after up conditioning or down conditioning of the H reflex. So this is up conditioning. And what happens is that the axon initial segment gets a little farther from the soma and it gets longer. And modeling studies predict that these changes will lower the threshold, will make it more likely that the action potential fires. So this could explain the increase in the size of the H reflex. In any case, it shows that there's very striking change occurring here in this really the final gatekeeper for any action. Similarly, what you see with down conditioning is very different. There's increased GABA input, inhibitory input to the soma and to the axon initial segment. Anchorin G, which is associated with sodium channels, decreases. PP38, which interiorizes sodium channels, increases, and the H reflex gets smaller. So here, basically you see that, and again, this change could explain the smaller H reflex. But in any case, what these data show is that here, all we've done is operant condition this stupid little reflex, the simplest behavior, and it greatly changes the final gatekeeper for this muscle, for this motor neuron, and thus for the muscle. Thus, it affects every behavior that uses this muscle. Every behavior, every network that uses this muscle has to adjust because of what we did here. So this is a very striking example of really the need for a negotiated equilibrium. They have to adjust so that they can continue to perform because we've done this very ostensibly very simple behavioral change. We produced it. So the CNS substrate of a skill is a distributed network of neurons and synapses that produces a skill and changes continually to maintain it. This was presaged by Bernstein in 1967 when he said that behaviors are biodynamic structures that live and develop. That's what this network is. Together, these networks maintain a negotiated equilibrium in the central nervous system. It became clear that the networks need a name that implies their special properties. And I worked over a number of years, actually during the pandemic, with long distance with Adam Commissar, who's a classical scholar at Hebrew Union College in Cincinnati. And it turned out there was a perfect ancient Greek concept that really conveyed our concept of what this network is and does. We're calling them hexers from the ancient Greek word hexus. It was a word originally defined by Aristotle and then changed some by the Stoics after him. It very much fits the concept of the CNS basis of a skill, and the OR ending implies that this is an active agent. It's an actor, it's an effector. So we're calling these networks hexers. These are hexers. This is our current understanding of the hexer for finger flexion sequence learning and for spinal reflex conditioning. This is a concept of a very brief rendition of negotiation among hexers. Here's a naive nervous system with a whole bunch of neuronal and synaptic properties that can change early in life. We acquire flexion withdrawal reflexes, and a set of these properties change to produce appropriate flexion withdrawal. A little later, we learn to walk, and an overlapping set of properties change to produce walking. And these guys here have to change also because these guys have changed and they still need to produce flexion withdrawal. So this is the result of a negotiation. Later on in life, a skill may be acquired, such as discus throwing, and this will lead to further changes. And this one is particularly interesting because discus throwing is These are symmetrical actions, symmetrical skills, flexion, withdrawal, walking. The two sides of the nervous system can be pretty identical and to produce that. However, discus throwing is very asymmetrical. So there's going to be a very complex negotiation at every level from the brain to the spinal cord so that discus throwing can be mastered, and at the same time, flexion withdrawal reflexes and walking can be maintained. The implication, and we're now doing experiments to try to demonstrate that, the implication is that flexion withdrawal will still be maintained, but it'll be performed differently on the two sides of the nervous system. And similarly with walking, there will be asymmetries produced by the negotiation that there'll be activity asymmetries, but walking will still be symmetrical and satisfactory because of the acquisition of this new discus throwing task. So the CNS is in a state of negotiated equilibrium. Here's an example of what happened, of a very simple piece of a negotiation. Here's a rat walking along on a treadmill. You see the right and left activity in the soleus muscle as the rat walks along. We can change the size of the reflex by up training or down training the H reflex, and this affects the soleus burst during locomotion. It gets smaller with down conditioning, it gets bigger with up conditioning on the side that we trained. But walking is not impaired. Walking stays symmetrical. EMG and kinematic changes maintain symmetry in hip height. Here you see the joint angles as the rat's walking forward. The rat's head is up here, hip, knee, and ankle. This is for a smaller reflex. This is before conditioning. If all that happened was that the soleus activity, soleus burst got smaller, the ankle would become more dorsiflexed here, and the hip would be lower, and the animal would be twisted as it walked along. It would be tilted. But that's not what happens. There's a compensatory increase in the hip angle. So as a result, the hips stay even. The animal remains symmetrical. The kinematics are a little different on the two sides. And conversely, with a larger reflex, if this was all that happened, the hip would be high on that side and the animal would be tilted to the other direction. Here, but this decreases and the hip stays symmetrical. The key feature is maintained. The old locomotion hexer adapts to the new H reflex hexer. How does this understanding affect rehabilitation? The central nervous system is not hardwired. It is continually changing. Whether healthy or damaged, it is continually changing. In the healthy central nervous system, ongoing hexer adaptations ensure negotiated equilibrium. In contrast, the chronically damaged central nervous system is a contentious, chaotic environment. We can normally think about it as a stable state. It's not. They have damaged hexers that are trying to repair themselves, compensatory hexers that are trying to take over, primitive hexers that come out. You get initial behaviors or synergies that occur early in development and normally get suppressed in normal development, which can come out again after a stroke, for example. You get diaschesic processes, which are processes in which damage in one area leads to changes in the areas connected to it. You get ipsy-contra influences. After a stroke, is the intact hemisphere good or bad? And the answer is probably yes, and it probably varies from person to person. It has influences that can be good or bad in terms of recovery and other life events that occur. Here's an example from a really nice study by Murata, now about 15 years ago, really showing competition between a normal and a compensatory hexer. This is a situation in which a monkey has a lesion made, a small lesion made to the area of sensory motor cortex that controls finger movements. So this is basically a small stroke. This is the normal precision grip that the monkey would use to pick up a food pellet. after the stroke is induced, there are more, and the animal starts to recover, you get alternate grips that occur. For example, the finger will grab the pellet against the metacarpophalangeal joint or against the interphalangeal joint here. There will be less ideal ways of picking up the pellet. So you have a grip, first you have a grip deficit after the stroke, then you get alternate grips, this incorrect way of picking it up. It's somewhat effective, but it's not ideal. And this is the normal precision grip. So if the animal does not practice afterwards, this is the days after the lesion. Here you have before the lesion, when the animal is using this precision grip very well. Afterwards, it can't do anything for a while. And then it starts using these two more primitive, less effective ways of gripping. And there's an area where you actually see some of this come out, but then it gets suppressed. And you wind up with basically this less than satisfactory grip. Alternatively, if you have the animal practice every day throughout this period, what happens is that the grip eventually comes out and eventually triumphs. So you wind up with, again, mostly the precision grip. There is a competition that occurs here between the less than ideal grip and the precision grip. And you can see that here in success rate. So this is what happens when the animal doesn't practice. It sort of comes up to this alternate grip level, which is better, but is not ideal, versus here where it starts up this way, and then there's a period where it actually gets worse. There's actual competition going on here. The success rate goes down, and ultimately, the good guy wins as a result of practice. But this is an interaction that's occurring. This is a competition occurring here. So the chronically damaged nervous system is probably best described by the 19th century English poet Matthew Arnold in the poem Dover Beach. It's really like a darkling plain swept by confused alarms of struggle and flight, where ignorant armies clash by night. That's what's happening here. That's the ignorant armies are competing here. And in this case, at least, the good guy wins. Thus, basically, the point here is that rehab therapy is not going into a stable, damaged state. It's entering a complex, active, and perhaps hostile environment. So the goal of rehabilitation in this new paradigm is to enable damaged hexers to repair themselves and reestablish a negotiated equilibrium in which each hexer can once again maintain the key features of its behavior. The mainstay of standard rehabilitation is skill-specific practice. Skill-specific practice has two limitations. First, in the adult CNS, as we know, plasticity is there, but it's limited. So damaged pathways are not going to automatically regenerate themselves. The solution is to develop methods to produce more generalized plasticity. That's one solution to the limited plasticity, to increase that plasticity. Hexers then have more options during practice. The other is less recognized, but is also there. The available plasticity is not optimized. There are lots of ways. These actions, such as locomotion, such as talking, are very complex. There are many places where plasticity could occur. The solution is methods to produce targeted plasticity. Normally the plasticity is not optimized. There's no little person in the head who can look at all the availability and say, oh, we want plasticity there. We want plasticity at this place, and then at this place, and then at this place to this amount. That doesn't happen. There's no overall oversight. So the solution is methods to produce very targeted plasticity and guide the Hexer adaptations during practice. This leads to the overall concept of triple strategy rehabilitation protocols. And that's illustrated here. This is a healthy person. This is the locomotion Hexer, in which you have neuronal and synaptic properties that are all in good shape here to produce normal locomotion. You have a CNS injury, such as a spinal cord injury. Some of these structures are destroyed, others become abnormal, and as a result, walking is much impaired. Generalized plasticity methods will restore these missing parts. Targeted plasticity will target plasticity to a crucial point to produce more normal function. And the result, when you have skill-specific practice, is a recovered or largely or partially recovered nervous system, in which you have some areas that are exactly as they were before, some structures that are different, but they're satisfactory, so that you still can produce the key features. This, for example, this could be a method that regenerates spinal cord pathways and makes new connections. And then the targeted plasticity shapes those connections so that they function properly. Or it could, in targeted plasticity, it could decrease a hyperactive reflex to allow practice to occur. You can have a hyperactive reflex that produces clonus or foot drop so that a person cannot practice locomotion. If you can remove that, satisfactory practice can occur and much wider change can occur. So these are examples of generalized plasticity. One is Schwann cell transplantation. Schwann cells myelinate peripheral nerves, and it's been shown that when Schwann cells are injected in the central nervous system, they can increase the survival of damaged tissue, and they can become functional. So you get spared, you an increase in the amount of spared nervous tissue. Walking improves, the walking scores improve more with the Schwann cells, and a function such as reach and grasp improves more in rats when the Schwann cells are injected into the spinal cord. This is another method. This is the intracellular sigma peptide. This is a method that was developed over many years by Jerry Silver at Case Western Reserve University. He very sadly died about a week ago, but his achievement is going forward. It's now being tested in people as well. But the idea is that chondroitin sulfate proteoglycans, after an injury, prevent regeneration of axons. They block axon regeneration by binding with a receptor. The intercellular sigma peptide internally prevents that receptor from being effective and leads to much improved regeneration. These are serotonergic neurons in that situation, regenerating in an injured spinal cord. So, and you can see, now this works. It works in some rats and it doesn't work in others, and that's something that's now under investigation. But in any case, when it works, you can get improvement in locomotion because you now allow regeneration to occur. So this is another one of the new techniques that's being developed to produce generalized plasticity so that the hexers can then shape. So the other strategy is to target beneficial plasticity. We've done that with reflex operant conditioning. This shows, for example, that you can train an H reflex to be smaller in someone who has, after a spinal cord injury. You can decrease the size of the H reflex to reduce spasticity, reduce foot drop, reduce clonus, And for example, here you see in that situation, this work was done largely by Aiko Thompson. So the soleus EMG, this is the soleus muscle activity during stance and swing. And this is normally in a healthy person, there would be activity during stance and no activity during swing. But this is abnormal activity occurring during swing. Afterwards, after down conditioning of the H reflex, that activity is largely gone, and that remains gone 6 months later. And the important point here is that the improvement in function that occurs is not produced just or even probably mainly by the exact targeted plasticity. This leads to better practice and much wider plasticity. So by down conditioning the H reflex, the abnormally active H reflex in the more damaged leg after a spinal cord injury, activity improves in the muscles of both legs so that the soleus burst gets better. Remember, we down conditioned the H reflex, but it allows the soleus H reflex, the soleus burst, to get better in the muscles of both legs. And the tibialis anterior activity also becomes more normal. The abnormal activity during stance goes away, and the tibialis anterior activity becomes more normal. So you get widespread changes that are enabled and essentially triggered by this guided plasticity, which eliminates a crucial deficit. And as a result, people now can walk about 60% faster, and they walk more symmetrically. Here you see the onset of right and left stance in someone with a spinal cord injury. And this is when, this is halfway between here and here, say halfway between right stances. This is when left stance the onset of left stance should occur, and it's occurring late. Afterwards, it occurs on time. So we've restored symmetry and speed to locomotion from this triggered plasticity, from this targeted plasticity. This is an example of that. This is a man in which the H reflex was down conditioned 5 years after a spinal cord injury. Here he is walking before H reflex conditioning. He has a walker with stoppers on it. He has someone standing next to him. He has a wheelchair behind him. Here he is afterwards. He's now got tennis balls here. He doesn't have a wheelchair behind him. He doesn't need someone standing next to him. And here he is walking along. He's going about 3 times as fast, and his walking in general is better. It's not just this small reflex that we've changed. There's much wider change that has occurred. So now he's done with the 10 meters, and here he's still going. This is a person after 2.5 years after a stroke. This is another kind of operant conditioning which increased the extensor carpi radialis motor evoked potential, again by operant conditioning. It's showing her performance on the box and blocks task. So the number she's managed to move from one side to another is shown here. She's going 2 or 3 times as fast afterwards. Her arm is moving more normally. It's not normal, but it's moving more normally than it was before. She's a lot more effective. This is another way of targeting it. This is developed by Monica Perez's lab, in which you use Hebbian plasticity to strengthen the corticospinal connection in the spinal cord. And that's paired associative stimulation. This shows the increase in the MEP of the biceps and the abductor pollicis brevis. When you have sham PAS, that does not occur. It's associated with increase in the size of the maximum voluntary contraction and increase in performance. The time to perform a set of functions is much decreased. This is illustrated here. This is a man, again, 5 years after a spinal cord injury, walking. This is before paired associative stimulation. This again is the Shirley Ryan Ability Lab, Monica Perez's group. And here he is after 40 sessions. This again is an hour-long session. This is all non-invasive. So operant conditioning, paired associative stimulation, this is all non-invasive. Walking much more effectively. And here he is after 80 sessions without the harness. This is 5 years after the injury. So these are several of the non-invasive protocols that are now coming along and being developed. They're now in the process of being converted into methods that can be used more, being automated and put in systems that would be much more robustly suitable for widespread clinical use. There are a variety of protocols, a large variety that could be developed. The evaluation has very specific requirements, appropriate evaluation. Controlled trials are essential, both to compare the new protocol to state-of-the-art methods and to verify the presumed mechanism, and to detect individual specific negative effects. There's a real problem with minimal risk protocols that are approved without controlled trials. They may actually be inferior to state-of-the-art. They may be harmful for some patients, and they distract attention and funds from validated protocols. So they're really therapeutic noise. Without controlled trials, you can't really know if these are any good, but they really can clog up the field, and they can take they can distract attention and funds from actually validated protocols. This is very important, and it's going to be an ongoing problem for a while. So in summary, there's lifelong CNS plasticity, which we didn't know 50 years ago. The challenge is to use it to restore impaired skills. The substrate of a skill is distributed from brain to spinal cord. It's now called the hexer. Hexers share the CNS. They affect each other. Each one is changing continually to maintain the key features of its skill. The hexers keep the CNS in a negotiated equilibrium that serves them all. Effective rehabilitation helps damaged hexers restore themselves and regain a satisfactory negotiated equilibrium. There are two new strategies. One is methods to produce more generalized plasticity, and the other is produce targeted plasticity. In combination with skill-specific practice, both new strategies produce widespread beneficial plasticity that enhances recovery. These are triple strategy protocols. Controlled trials are essential, mechanistic controlled trials, and state-of-the-art controlled trials. We need, hopefully, to identify biomarkers that can enable us to predict and help select patient-specific protocols, predict the outcomes. And another big push in this and possibility is that a lot of these can be made home-based protocols, so they can be much more widely and economically used to help many more people. So this is hopefully where we're going in the next decades, but we're just really at the beginning of this, and there's a great deal to be done. It's a very exciting as well as frustrating time, just because there's so much to do and there's so many possibilities. There have been many people in our group who have been involved in this over the years. Here at our National Center, we've had support from NIH, from the Department of Veterans Affairs, and from other organizations. Thank you very much. I look forward to your questions and comments.
Thank you very much, Jon. Virtual round of applause for our speaker. And we're officially open for questions. And we already have one. Simis, go ahead.
Right, thank you very much. Thank you, John, for this extremely interesting talk. I was wondering about the time scale of this rebalancing of flexors that you're mentioning. And especially, I was thinking that this cannot be too fast, which means when people acquire a very, let's say, precise new skill, let's say dribbling in football, then this should probably lead, at least temporarily, to some other skill becoming worse, at least for the time being. At least this rebalancing can take place again. So have you ever seen that experimentally? Let's say if we take Lionel Messi and we teach him to dance very well, will his dribbling skills become worse, at least for a couple of weeks or so?
Yeah, that's a very good question. Presumably there will be these interactions, and the question is, what's the time scale in which there will be a response? I mean, I think the important thing to realize is that, we talked about, I mean, the easy way to think about this is that, okay, every time you do the skill, you adjust it, but that really is not enough to explain what happens. You can ride a bike not for 50 years and you can still do it reasonably well, and your body's completely different and you've learned all kinds of new things, etc. So it has to be that they are continually adjusting themselves even when they're not active, that there is interaction and that this is ongoing continually. I mean, I think that is a good deal of what spontaneous activity is in the nervous system. This is continual negotiation and continual optimization, or not necessarily optimization, but continually adjustment of all the hexers. Obviously, actually practicing it makes it better, but they have to be essentially learning from each other. In terms of interfering with each other, I mean, we're trying to look at that now, and Russ Hardesty and Helia Mojtababi in our lab are looking at that. We're trying to look at what happens when people learn to practice walking forward with one leg and backward with the other 3 times a week over months. What does that do to normal locomotion? What does that do to reflexes? We're just at the beginning of that. We have a lot of data. We haven't analyzed it yet, but normal locomotion is presumably going to change how it's produced, but we're looking at that. But yeah, there would be probably some at least transient impairments, but I think they'd probably be relatively minor in normal situations.
I have a comment that would pertain to that, John, if I may.
Yes, but
When listening to your talk and your hexars, it was reminding me a lot of the idea of predictive coding, where you have distributed neural networks throughout the nervous system, as you pointed out, with movement, and then at each level there are prediction error neurons. So you can imagine that there's an overall schema of what the overall movement should look like, what it should be like, and then you have and I think you can see this in the cerebellum, where you have descending inputs of what this movement would look like. And then you have inputs, sensory inputs, that are what the actual movement was. And if there's any discrepancy, you get activation of these prediction error neurons, which causes the network to change so that it reduces that prediction error. And I think that whole idea, we are constantly having this evaluation of prediction error neurons in the system to see whether or not the movement is being done correctly, can be adjusted moment to moment.
yeah. Yes, I mean, well, clearly, I hope can you send me some summary of I mean, that sounds very relevant, very reasonable.
Yeah, right. And I have another comment is your emphasis, right? I mean, this has been your history. I know when I worked for you many decades ago, or centuries, or whatever it's been. I remember, the focus was on spinal cord injury and how to rehab it. But listening to your talk, I'm thinking there's a whole other area where this could be applied, and that's where the nervous system has been reorganized incorrectly. That's resulting in some disorder, and one that comes to mind is chronic pain. It's thought that some types of chronic pain are just the nervous system now is now responding incorrectly to non-painful inputs and creating this chronic pain. And if it seems like your techniques should be applied to that problem. And there you've got a normal nervous, well, it's not a normal nervous system in terms of wiring, but it's an intact nervous system and should perhaps be more amenable to change.
Yes. Yeah, that's very apropos also. I mean, Disha Gupta in our lab is very interested in addressing pain in this way. And Jody Brangaccio is also involved in that. But the idea using these methods, for example, in amputees with phantom limb pain, to try to reduce it. But yeah, this is very, very relevant. In terms of checking errors, I mean, one of the issues that come, I mean, there are lots of issues with hexers, but how are the key features preserved? Clearly, this addresses the issue of, the plasticity stability dilemma, which affects neural networks, but it really isn't. In this paradigm, it's not really a dilemma because the plasticity of the hexer ensures the stability of if the memory is the key features, the plasticity of the hexer ensures the stability of the memory. But how exactly that would be preserved? Would it be, you could imagine symmetry in which you had two sides that had to be operating symmetrically for a symmetry key feature, but there's essentially very little, certainly I have very little understanding about how this would actively be stored, and how these entities would be basically, they need to be supported in this environment. So there needs to be some sort of basically, I guess, ongoing reward, which is essentially the energy that guides them. But in any case, yeah, both your things you mentioned are definitely relevant. And it's good to see you.
Good seeing you, Todd.
Go with Fabian.
Can you hear me?
Yes.
So thank you. Hello, Jonathan. Thank you very much for the talk. I guess I have a question that is also related to the previous question. So my initial question was like, how do you think these key features are identified and stored somehow to be replayed? But you just mentioned that indeed it's probably not something we have a good understanding of. But I was wondering, these XOR, I guess this memory of the key features that they need to maintain, they are kind of part of the XOR. And for instance, in the case of stroke patients, maybe the target key features can also be damaged as part of the lesion, right? So maybe it's not only the network itself that is damaged, but also the target key features that they need to maintain, and maybe identifying where they are stored or how they are stored in some way could also be a way to maybe help the XORs to self-repair, for instance. I don't know if that's something that makes sense or...
Yes. Yeah. I mean, yes. I mean, you're at least as far along on this as I am, but yes. There is presumably the practice, the function of practice has to, with no situations, would have to be would contribute to restoring those, but I don't know. It's a very reasonable, there's a lot of this. I mean, that the way you phrase that is something I had not thought of before, and something I will definitely be thinking about. But yes, there are a lot of questions that this raises that are not straightforward, or are not necessarily not straightforward, but are not well defined at this point. And when you think of hexers, the very limited kinds of hexers that I've talked about, like what we do with the H reflex, or locomotion as a hexer, or what defines one hexer as opposed to another. it becomes murkier, but that will happen in any sort of paradigm that tries to address the more complex sorts of skills.
And maybe if I may, Anna, another question. So this question was on the first property of the XOR, that they maintain these key features. And the second one was the negotiation between XORs. And you mentioned that it's this negotiated equilibrium. But you also showed this example on the monkeys in which, in the end, the good XOR wins. So in that case, the negotiation was not an equilibrium, right? There was a winner. And so how do you think this negotiation occurs, and how can we push one XOR or the other, for instance? Is it like by doing this targeted plasticity, for instance, or do you see other mechanisms?
Yeah, I mean, clearly the amount of practice is important. I mean, in that case, in that Murata study, the only thing she had different was the amount of practice. The monkeys that got better practiced a lot, and the others basically weren't using it at all. It was basically, at least that those finger movements were essentially immobilized or very little used. So that's important. And I mean, that's really a compensatory. When we talk about negotiation, I mean, I guess that's an antagonistic sort of negotiation, in which essentially one winds up getting used a lot more than the other. But yeah, that's something else that, how many hexers do you really need, or whether clearly both of them are not necessary, probably. The question is, and that becomes another issue, which frankly I hadn't thought of previously. I mean, how do you decide whether you don't need this one, and what determines that? So
yeah, it's all intertwined, right? So that's tricky in the end. But
yeah.
Maybe just my last comment. But maybe that means when you do rehabilitation, traditionally we focus on the damaged XOR, but maybe that means we need to consider the other XORs as well to somehow steer the negotiation wherever we want to go. But again, it's probably very, very difficult to do in practice, but that would be the ideal, I guess.
Yeah, that's another reason for, I mean, the more we can get this out of the lab and out of the clinic even, and into home use, things will get interdigitated better and will come back better. I mean, I know there's increasing attention with that. What you do in the clinic doesn't necessarily translate. And if you train on one task, does it translate out into its use in the real world, into related tasks, et cetera? But those are really, I mean, it's good that we're actually coming up against those issues now, but they're definitely there.
Thank you very much.
Thank you, Fabian. And last question, Guzman.
Thank you very much. Can you hear me well?
Yes.
Yeah.
Yes.
Thank you, Professor Wolpaw. I'm Mexican. I'm an engineer by formation, but I work in neurorehabilitation in a small clinic in Mexico. But as part of my thesis, we started working with Harvard and at the University of Toronto, and we are trying to develop a concept called prohabilitation, which is much more like extensions of what we already have as human beings. Two examples. If we have a sixth finger, for example, with a robotic or mechanical device, do you believe this can change the patterns of the hexors in your analysis, in the patterns that we can observe? That will be like my first question. The second one is I wrote in my thesis in Spanish. I'm going to say it like that because it makes more sense. That we are somas and we are semas. Somos somas y somos semas. So this is connected with the somatic side and language. And we start analyzing this in the brain when we were asked by a company how to create new devices. For example, in a computer interaction, we have a keyboard, we have a mouse, and we have a screen. but what if we could integrate these three devices in one single device, like a sphere, no? I mean, you grab a sphere with the two hands, you tap in the surface of the sphere, you rotate the sphere, and this is a direct brain interaction. We can call it like that. Do you think these hectors can change or evolution as patterns in the future? And if this can also reflect in economic theory, for example, because it's going to be a completely different way to negotiate in extensions that we cannot realize yet. So this is just experimental for the explorer, but I find it fascinating your talk. So I really appreciate it.
Yeah, I mean that was I was really interested in what you're doing. I mean, it gets into the issue. The first thing I think about is, do you do it, when you have this new device, is it a new hexer or is it an old one adapting to it? And that's something that's going to happen too, because initially when you try something new problem comes up, you apply what you have. So you're going to try to use the hex, presumably the existing hexers are going to try to handle this issue, and maybe they can, but maybe they can't, and maybe eventually you wind up with something new. And the way I define it, define what's a new hexer and what's a changed hexer, et cetera, is really in terms of the key features. So that like ballet overlaps obviously a lot with locomotion, but it has other features because it has to be attendant to the music, for example, and it has to be to certain kinds of positioning that are important key features in ballet. And you can walk without paying attention to that, and your walking is going to be satisfactory, but it's not going to be good ballet. So it's an overlapping but different skill, but different hexer. So yeah, that with the clearly that would produce things, that would produce something new, what you're doing, and it would be the amount of practice you do with it, and do you do it instead of using the mouse and the keyboard at all? Does it take over? That's very interesting. That's neat.
Thank you.
Okay, no more questions. In which case, thank you, Jon, again. That was really good, really interesting talk. Our next one will be sometime in February. Everyone will receive the details as usual. But yes, final thank you to Jon for his time and for joining us very early from his side of the world.
Well,
it took us a while to get you scheduled, but I'm very happy that we managed.
Okay. Yeah, thank you. It was fun.
Thanks. Thank you.
Yeah. Bye-bye.
See you else in a month.